Good morning, sir. Good morning, guys. This is Jade Santos from BSET AC. Before I start my report, konting disclaimer lang, hindi ko po kayang i-further explain ang aking topic since konti lang po ang time na binigay para i-present ng aming um, report. So, all insects have complete digestive system. This means that food processing occurs within a tube-like enclosure, the alimentary canal, running lengthwise through the body from the mouth to anus. Ingested food usually travels in only one direction. So my report is insect absorption. What is absorption? Absorption refers to the uptake of the compounds in the gut lumen across the gut wall to the hemicell. At the cellular level, most absorption is transcellular, meaning that the compound is transported across the apical cell membrane of gut epithelial cells, transits these cells, and is exported at the basal membrane. Paracellular transport, example between cell of the gut epithelium, is generally uncommon. An insect uses its digestive system to extract nutrients and other substances from food it consumes, but most of this food is ingested in form of macromolecules and other complex substances such as proteins, polysaccharides, fats, nucleic acid, etc., which must be broken down by catabolic reaction into smaller molecules. Example, amino acid, simple sugar, etc before being used by a cell of the body for the energy, growth of, or reproduction of this insect. This is breakdown process known as digestion. Next is the transport of amino acid and protein. Most research on the absorption of sugar and amino acid has focused on their passage across the apical membrane of enterocytes. Multiple sugar and amino acid transporters have been identified in various insects. Some transporters media active transport. Example, transport independent on energy to move a substance against its concentration gradient. Next one is the mediate passive transport. Transport not requiring energy and dependent on higher concentration in the gut lumen than in the enterocyte cells. Passive transport can be promoted by the metabolism of transported compound into the enterocytes, thereby maintaining a steep concentration gradient across the apical membrane. The midgut amino acid transporters in most insects that have been studied belong to the sodium ion coupled neurotransmitter and amino acid importer family known as family SLC6. Multiple transporters are expressed with overlapping specificities for amino acid. Some are very specific. Example is the N86 and N88. In the distal midgut of the mosquito Anopheles gambiae transport just aromatic amino acids. Other SLCs 6 transporter have a very broad range, notably the neutral amino acid transporter in Drosophila DMN86 can mediate the transport of most amino acid apart from lysine, arginine, aspartate, and glutamate. And remarkably, it can also take up the isomers for several amino acids. This capability can be linked to the abundance of the amino acids in the cell wall of bacteria, which are important component for natural diet of Drosophila species. DMN86 is an active transporter capable of mediating uptake against the concentration gradient. Exceptionally, amino acid transport in the midgut of larval Lepidoptera is coupled to potassium ions and not and sodium ions. Next one is the transport of sugar. Carbohydrates are absorbed mainly in monosaccharide sugar across the midgut epithelium, but the molecular basis of sugar transport has been little studied. There is general expectation of parallels with glucose absorption in a mammalian intestine, in which transport is transcellular and mediated by the sodium glucose in which transporter is transcellular and mediated by the sodium or glucose co-transporter SGLT1, a member of a sodium solute symporter family, and two facilitative glucose transporter is the GLUT2 and GLUT5, both member of major facilitator family superfamily MFS. Consistent with this expectation, glucose transport across the midgut of the hymenopteran parasite Aphidius irving is mediated by the SGLT1 like transporter on the apical membrane together with a GLUT2 like transporter on both the apical and basolateral membranes of the enterocytes, and the second passive transporter similar to GLUT5 is implicated in fructose uptake. This condition is not, however, universal among insects. For example, genome annotation of the pea apid Acritosiphon physum revealed no sodium or solute importer with plausible specificity of sugar, but 29 candidate sugar transporters in the MFS family equivalent to the GLUT. These included an abundantly expressed gene APST3, a hexose uniporter with specificity of glucose and fructose in the distal midgut. The metabolism of absorbed sugar, both as respiratory fuel and in trehalose and glycogen synthesis, may also promote sugar absorption. Next one is the transport of lipids and related compounds. The product of lipid digestion include free fatty acid, glycerol, mono and diacylglycerols, and lipophospholipid. These compounds can move relatively free across membrane and they are absorbed principally across the midgut epithelium. Although absorption in the foregut, example, the crop of cockroach Peripalanet Netta americana can also occur. Two-factor promotes lipid absorption. The first is emulsification, which assists their diffusion across the aqueous boundary layer to contact the apical membrane of gut epithelial cells. Insects lack the bile salts that are crucial for emulsification in the vertebrate gut. This incidentally is why the insect-neutral lephases do not have cholephase cofactor, which is required by the pancreatic lephase of vertebrate to hydrolyze the bile salt covered lipid in vertebrate gut. Instead, emulsification in the insect gut is promoted by formation of fatty acid, amino acid complexes, and glycolipid complexes, and the formation of mixtures of fatty acid and lipophospholipid. Next one, the second one is the promoting absorption is the resynthesis of lipid from absorbed compound in the enterocytes. This metabolic activity maintains a steep concentration gradient promote uptake. In all insects studied to date, the enterocyte synthesis 1 to 2 diacylglycerols, triacylglycerols, and phospholipid. Lipids can also be generated in enterocyte from absorbed glucose, galactose, or other sugars. Lipids are exported Lipids are exported from enterocytes to the hemolymph predominantly as DAGs and are transported to other tissues bound to the protein lefoprin. Lefoprin has also been implicated in the transport of hydrocarbon carotenoids, sterols, and phospholipids. Sterols appear to be absorbed unchanged, but in some caterpillars, sterols are esterified in the gut cell. Absorption of inorganic ion. Sodium to move from the gut lumen into the hemolymph of a glucose, it must move against the electrochemical gradient. Energy for this active movement is provided by the ATPase pump in the apical plasma membranes of the anterior ceca, and the bi-major sodium-potassium exchange pumps in the basal plasma membranes of the rectal cells. In potassium, on the other hand, probably moves passively down its electrochemical gradient into the hemocell. The electrochemical gradient for chloride and calcium ions are less extreme. In the rectum, chloride ions are actively removed from the lumen by pumps in the apical membranes of the cell and pass passively from the cells to hemolymph. Calcium is actively moved from gut lumen to the hemocell by cell in the gut. Absorption of water. There are two major water absorbing zones in the insect gut. One in the midgut, where water is absorbed from food, and the other in the rectum, where water is absorbed from the feces before they exit in the body. Absorption in the rectum is the key component of regulation of body water. Water absorption in the midgut serves to concentrate the food, enhancing body the efficiency of digestion and the maintenance of concentration gradient favorable to the absorption of nutrients across the wall of the gut. It also creates water flow, which are important in nutrient absorption and perhaps enzyme conservation. Specifically, absorption and proximal midgut, especially the midgut ceca, where this structure occur, drives the forward flow of water in the endoperistrophic space, carrying forward the product of digestion. The water may be derived directly from food, as well fed grasshopper or drone forward as it leaves Malpighian tubules, as in cockroaches, in larval Diptera, and perhaps in some other insects. The forward movement occurs in the ectopeteropic space while fluid in the endoferrotrophic space move backward. This countercurrent probably improves the efficiency of utilization of digestive material in gut. Water absorption depends on the establishment of osmotic gradient across the epithelium, such as water passed from zone or low solute concentration to high solute concentration. In many insects, the flux of water from gut lumen to the hemolymph is based on the accumulation of sodium and potassium ions in the extracellular space between epithelial cells, which has only a few openings of hemolymph. Water is thus drawn through or between the cells from the gut lumen into the extracellular space. This creates a hydrostatic pressure which forces water out through the openings of hemolymph. This process is energetically very demanding and is supported by a large number of mitochondria close to the cell membrane. Water absorption from the midgut often occur in localized areas. In cockroaches, grasshopper, and the larvae of some flies, mosquito, and Sciuridae, it occurs in the midgut ceca, while in blood-sucking insect, water is removed from the stored meal in an anterior midgut. Deep invagination of the basal plasma membrane, which may extend more than half toward the apex of the cell, are closely associated with a large number of mitochondria. These provide the energy by which potassium or sodium in case of rondius is actively pumped into the. So that is all. Thank you. Here is my reference.